starting with the Human Genome Project, the goal was to sequence DNA and find the sequence of the human genome. And we wanted to sequence each individual chromosome and sequence them as well. And chromosome 1 was the last one to be identified and covered. Now, for the Human Genome Project, we use sequencing machines that use the deoxy chain termination method that we learned in Chapter 20. Now, going deeper into it, we use next-gen sequencing in order to create high-throughput sequencing, which basically speeds up sequencing from these old methods that we used. So the old methods that we used were methodical sequencing and whole genome shotgun sequencing. Whole genome shotgun sequencing takes DNA, clones it, and then cuts the DNA into individual fragments, and then sequences the DNA, and then reassembles it into one clean single sequence. Now, all these methods were used, but now next-gen sequencing has been introduced, which speeds up sequencing and makes things a lot faster for sequencing our genome.
Genomics is a next-generation sequencing technique in which DNA from an entire community of species, or a metagenome, is collected from an environmental sample and sequenced. Although it is currently in early implementation, metagenomics focuses on non-eukaryotic microbes. There is no doubt that its concepts and methods will ultimately transform all of biology.
So in just this way, genomics, a science developed to aid the advancement of biomedicine and the understanding of our own species, transformed the science of organisms and the application of all the sciences in epidemiology, clinical microbiology, and many other fields. For instance, in the field of biomedical sciences, which is in the realm of health and disease, in which individuals and populations and the development of novel diagnostics and treatment strategies based on the knowledge.
The other field that we'd like to talk about is biodefense and microbial forensics. Here we can develop more effective vaccines and therapeutics against potential bioterror agents, and the development of genomic biosensors to monitor microbial ecosystems for known and potential pathogens.
The NCBI website, a co-product of the National Institute of Health and the National Library of Medicine, is extremely important for the use of bioinformatics. It contains a gene bank that includes more than 213 billion base pairs, or nucleotides.
Under the website The Blast, basic logic alignment search tool that compares sequences through base pair comparison. Besides nucleotide sequences, one can also compare amino acid sequences. Ultimately allows for better understanding, both visually and structurally, speaking of our genome. Scientists aim to identify all protein coding genomes and determine shape and therefore function in a method called gene annotation. We will delve into that part further.
There are 3 lines of evidence of gene annotation. First is pattern, looking for patterns indicating the presence of genes through sequences. ESDs, which are also known as express sequence tags, can be from the complementary DNA sequences online. If that doesn't work, then we have to compare identified genes of other species to see a possible link between the two. The DNA varies amongst species, so scientists might use that for the amino acid sequences, and then also analyzing biochemical studies. So although if this gene is a gene that is new and for which the new information is needed, then we can just turn towards biochemical studies, and CRISPR-Cas9 system is used where you can just knock out genes to see what happens.
We wanted to know more about the genome, so we decided to investigate using the ENCODE project. ENCODE is a long-term research project that we started to understand the functional elements of our genomes through experimental techniques using culture cells. Through this, we were finally able to learn more about the functional elements of our genome.
However, the ENCODE project did not have many clinical applications, so in order to find more clinical applications, we used the Roadmap Epigenomic project. The epigenome is the epigenetic of the entire genome. So basically what the project does, it compares the epigenome of stem cells of normal functioning cells to ones that are with autoimmune disease or cancers. Through this, we were able to make major discoveries, such as tumor growth.
The application of the Roadmap Epigenomics project has gone beyond oncology and drugs.
For example, there's a company called Johnson & Johnson that co-founded the formation of Rodent Therapeutics, which is biotech that functions in helping with creating a drug called histone deacetylase inhibitors, which will help with Alzheimer's disease.
In addition, there are also improvements in epigenomics of Crohn's disease and ulcerative colitis.